Okay, so now that we've talked about electrophoretic karyotypes, we can start to think about a whole new process of analyzing both the chromosomes and the genes that are found from doing these kinds of separations. And before we do that, just to make sure we understand, I have told you, and you have willingly accepted, that these bands that I'm showing you in this electrophoretic karyotype, say from a Chef gel, that they represent whole chromosomes from a particular fungal isolate, let's say. Okay, that's what I've said. Each of these bands represents a chromosome. But is that necessarily true? What's another possibility?
It could be something else that's negatively charged.
Well, it's going to be some piece of DNA, right? So it's going to be a piece of DNA. But I tell you that this is a whole chromosome. And actually, I'm going to do one other thing that will really kind of even emphasize this. Okay. So notice I've made these guys all the same thickness, and then I've got this one here.
Could be more than one chromosome?
Okay, so this could be more than one chromosome. What's another possibility for any of these bands?
Could it be less than one chromosome?
Meaning what?
Like a piece?
Yeah, it could be a piece. So is this a whole chromosome or a piece? And that's the question. I've told you that these are all chromosome sized and that they represent chromosomes. But before we work with that, before we move on with that, we need to be able to demonstrate that that's the case. And so how do we do that? Well, we're going to rely on the fact that chromosomes have certain components. So what does a eukaryotic chromosome have on it? Something that would be distinct, that every chromosome should have.
The centromere or centriole.
Okay, so the centromere. So each chromosome should have one centromere, and what else?
Telomere?
How many?
2.
Right, and 2 telomeres. Okay, so if each of these is a chromosome, then they should have a centromere and they should have 2 telomeres. Okay? So what we're going to do is start taking advantage of what we can do when we can physically manipulate DNA. And what we can do is, let's say that this is your chromosome that you're interested in. Let's say we take that chromosome out of the gel, and then we treat it with a restriction endonuclease. Remember, earlier in the semester we talked about restriction endonucleases as enzymes that can cut DNA, and they're specific about how they do it. So if we take this particular band and we cut it, and let's say we cut it with a restriction enzyme that cuts very infrequently. So if we cut a supposedly chromosome band with, let's say, not one. Not one, its recognition sequence is an 8 base pair recognition site. So what does the size of the recognition site that a restriction enzyme uses, what difference does that make in terms of how frequently it will cut? So let's say, sorry, go ahead.
The longer the recognition site, the less frequently it is likely to cut, because the less frequently that recognition site is to occur.
Exactly, exactly. So if we had a recognition site that was 4 base pairs, that should cut pretty frequently, especially in a chromosome-sized piece of DNA, you get a lot of pieces. But not one, which recognizes an 8 base pair recognition site, cuts infrequently. And there's some restriction enzymes. There's actually a restriction enzyme that's produced by yeast that cuts so infrequently that it never cuts the Eusulago-Matus genome, ever. There's no cuts, ever. We're not gonna use that because we actually do want at least 1 or 2 cuts. Okay, we want to get some cuts. So if we use this enzyme that cuts infrequently, then that enzyme, when we cut the band that we got, that we said was a chromosome, so if we cut with not 1, maybe we're gonna get 4, we know it's gonna be smaller than this, so let's say we get 4 bands in this lane here. Here's one lane, here's a new lane. Let's say we get 4 bands. Okay? Now, if we have a piece of DNA, let's use the telomere as an example. I'm not sure that there's a good one for this, but we know what telomere sequences of fungi look like in terms of nucleotide sequence. And so if we had that piece of DNA that represented the telomeres, we could take that and we could do an experiment known as southern hybridization, southern blotting. How many of you have heard of that? Both of you have, good. How many of you know what it means? Yeah, sort of, kind of. All right, so the idea is, if we take the DNA in the gel and we transfer it to a membrane, then the DNA in the pattern that is separated in the gel will show up on the membrane. And then what we can do is we can denature that DNA, and then we can take a DNA of interest that we know, and we can either radiolabel it or label it with some kind of color or fluorescence, and then allow that DNA to find its corresponding region in DNAs that we have on this gel. Okay, that's southern hybridization, DNA-DNA hybridization. And if we do that, taking the radioactive or fluorescently labeled telomeric sequence, and we hybridize it to the DNAs that we've cut and separated here, then we're gonna get some labeling. And we'll see if that shows up. So let's say that we do that and we get these two lighting up. Okay? So that's where the telomeric sequence, and we'll say that this is the label on the telomeric sequence. So you light up there and there. What does that tell us?
There are 2 telomeres.
That there are 2 telomeric sequences, and then these are the 2 parts of the chromosome where the telomeres are located. And we've been able to separate them by cutting the DNA with this restriction enzyme, and these 2 fragments represent those parts of the chromosome. And if we got that result, what would that tell us?
There's 2 chromosomes?
Well, the question is, how many chromosomes would there be if you get 2 bands that light up?
That there is 1 chromosome.
Yeah, there's 1 chromosome because each chromosome has 2 telomeres.
Oh, sorry, I was thinking centromere, my bad.
Yeah, no, you're right. Okay, so these represent the 2 ends of this 1 chromosome.
Gotcha.
Okay? Well, we can do that experiment a different way because, as was pointed out by somebody in the audience, that might be 1 chromosome. It might just be a bunch of it. Or you might have 2 copies of it. Or it may be more than 1 chromosome that's approximately the same size. We don't know what that is. Why is it thicker than the other ones? We don't know. But what we can do is an experiment that's similar to the one we just did, and that would be for us to cut this band out. Well, that would be to throw this out, but then we use this. There we go. Cut that band out, do the same kind of experiment with not 1. And then run that on the gel. And let's say in that experiment we got, so that's actually lower than that, we got several bands. And let's say that the result from that experiment was that we got that one labeled, this one labeled, this one labeled, and this one labeled. What would that tell us?
Now there's 2 chromosomes.
That's right. So this tells us that there are 2 chromosomes, whereas this tells us from the other experiment that there was 1 chromosome from the band that we looked at. Okay? Because of the number of telomeres that we're identifying when we take that band and cut it out. Okay? And just to make sure that you're following this, what if we had gotten only 3 bands that showed up with the label?
There's 1 in a piece.
Yeah, one of those is, there's a piece in here, not a full chromosome, in addition to a full chromosome. Okay? Yes, question?
What if there were, could there be 3 pieces?
You mean 3 chromosomes?
What if you got 3 labels,
Yes.
and instead of it being 1 chromosome in a piece, what
Yeah.
if it was just 3 pieces?
Oh, okay. Well, so that's a good question. Could you just have 3 fragments? That is a possibility. But I suppose, let me think of how you deal with that. I guess in that instance, you would want to see if you could further separate anything here. If you had a condition where if you just took this piece and ran it under different conditions, could you at least see some separation? Because that might at least allow you to work with the parts that you separated first. But yeah, good question. Okay. All right, now, so that's providing evidence that what we're working with is telling us that we're dealing with chromosome-sized pieces and that these are the chromosomes. Now, what this leads us to be able to do next is, let's say that this is 1 chromosome. And let's say that this, as we saw on the slide, represents the mating type chromosome from Microbacterium. And now what you want to do is you want to study the genes that are involved in mating type in Microbacterium. And the way you want to do it is by creating a gene library just from this chromosome, right? You could create a gene library from the entire genome to try and discover and study the mating process, but if you know that this is a chromosome associated with mating, or if we go back to the example with Nectria, if you know that that dispensable chromosome is that 1, well, you could make a gene library and you could do, which is what we're going to talk about now, in vitro genetic engineering and in vitro means
In the petri dish?
well in glass, right? So in vitro is like
Vitro, like
yeah,
glass.
Yeah, exactly. So in vitro means like in a test tube, right? So this is going to be in the test tube, as opposed to what we did at the beginning of the semester, or the first half of the semester where we did it in the organism itself, where the recombination was taking place. So this is going to be recombinant DNA technology. That originates in the test tube. With all of this technology that we're going to talk about from now through the rest of the semester, we do these things in the test tube, and then eventually they will get put into living organisms to see what the effects are. But the recombinant part is what we do in the test tube, and then that gets put into organisms to test what the effects are going to be. Okay? All right, so this again kind of sets up the process for why we might want to do an experiment like this. So let's say you're interested in the mating type system, and you know that this is a chromosome that's associated with one particular mating type versus another. You can seriously reduce the amount of work you're going to have to do if you just take that piece and make a little gene library out of that, and then you can study the pieces from that gene library and how they, what effects they might have on a particular function, okay? Now, not everybody in the audience is interested in mating type in fungi. I don't understand why that would be. Maybe you have some other interests. So let's imagine that some semi-irresponsible individuals decided to have a coronavirus party at their frat house. And after all of that debauchery, you happen to be wearing your protective, I don't know if anybody in frat houses, I don't know who does that, but you have your protective gear on, you've got your nice protective mask, and you just happen to be hunting, thinking about research projects, you happen to be hunting in the soil outside of the frat house after this party, which was eventually broken up, by the way, and you find an organism in the soil that is capable of breaking down alcohol. And there was a lot of alcohol that was being consumed at this party rather than being used for disinfecting various surfaces. And these bacteria that live in the soil outside of the frat house, they have developed a mechanism to become resistant to alcohol by being able to break it down. So even if you're not interested in mating type chromosomes and the genes on mating, genes responsible for mating in fungi, you might be interested in this particular organism because it is able to break down alcohol, and being of an entrepreneurial spirit, you are aware that if you can take that gene and work with it and produce it in large quantities, people will be able to drink large quantities of alcohol and not necessarily get drunk, I guess. You could probably sell that to frat houses, I'm thinking. So with this stimulus package that is being proposed, you might be able to get a grant to be able to study this particular organism, and then there could be a place for you in a pharmaceutical or biotech company when you are able to release this particular product to the market. Okay? So anyway, different ways to think about why we might want to do this. So what we're going to do now is we're going to talk about how you do this. And we're going to talk about it sort of the traditional way of doing cloning, and then as we proceed through the rest of the semester, we'll talk about some of the buzzwords that you've already heard. So you probably have heard of PCR, you may or may not know how it works. You may even have done it and don't really know how it works. You probably have heard of DNA sequencing, but you may or may not know how that works. We're going to talk about that. And then you've heard about targeted gene editing through CRISPR-Cas9. We're going to talk about how that works. But those are upcoming gatherings, not today's. Today's we're going to talk about sort of the traditional way of doing cloning. And what we'll do first is try to see if people understand what we mean by cloning. Okay, so what do we mean by cloning?
Duplicating DNA.
Okay, so it could be duplicating DNA, so this could refer to duplicating a piece of DNA. Typically, when we think about cloning, we think about isolating a particular thing of interest from the background in which it resides so that we can use it in particular. So when you say cloning, duplicating a particular piece of DNA, maybe what we want to do in that instance, and that's what we'll talk about in terms of methodology, is to separate that piece of DNA from its original source and put it into a situation where we can manipulate it and study it more easily. That would be what we talk about cloning as far as a piece of DNA. But when we use the term clone, what do we mean?
That duplicated thing, like whatever you've made a copy of.
Right, so an identical copy. And I'll put identical in quotation marks because oftentimes what we call a clone is not necessarily totally identical to what we started with. So in terms of some examples, if you look at the bacterial cells in a colony, those of you that have seen colonies of bacteria or colonies of yeast even on a plate, those colonies represent millions of theoretically identical cells. So each of those cells in the colony is theoretically a clone of every other cell in that colony. They're not 100% identical, because as we know, there can be subtle, random point mutations throughout a genome, even for individuals that are supposed to be identical. But essentially, overall, the cells in a bacterial colony or the cells in a yeast colony are identical cells, and we could call them clones. Another way you can get clones is if you take cells from a plant and do tissue culture. Theoretically, all of those cells in that plant then become identical genetically. Now, of course, if you take that tissue culture and cause it to differentiate into a plant, that plant will have resulted from a clonal population of cells. And then finally, the other thing that has really kind of been in the news for the last, 10 or so years, I'm trying to remember when Dolly the sheep, you guys old enough to remember Dolly the sheep? Yeah, Dolly the sheep. So this is an individual where you have a clone that's supposedly identical to the parental cells that were used to make it. Well, the way that that's done, in the case of Dolly, they wouldn't have been identical for a variety of reasons. So it's a clonal organism. One of the reasons that Dolly was not identical to her mother is because she had mitochondria that were different than those of her mother. Because what they did to make Dolly was to take the nucleus from, I think it was from mammary tissue of the mother, and they took the nucleus and put it into an egg cell that didn't have its nucleus, but it did have mitochondria. And then they caused that egg cell to divide with the nuclear material from Dolly's mother, and then that became Dolly. So that creature that we call Dolly the clone was not an identical clone by any means. And as we are aware, that creature had to develop from the fertilized, or in this case, manipulated egg all the way into a grown creature. And along the way, we experience different things during our period of growth and development that can alter the way that we ultimately develop. And so when we call Dolly a clone of her parental source, that's not true, not identical. But anyway, these are all concepts of clones. And now when we talk about cloning, what we're going to talk about, we're going to focus on cloning particular pieces of DNA. Okay? And the reason that we want to do that is because we want to be able to study them and perhaps manipulate them. So, for example, to produce large quantities of alcohol breakase for commercial purposes, or to find those parts of a particular chromosome that are responsible either for the ability to infect peas, or, in the case of a mating chromosome, the genes that are responsible for allowing the fungus to mate in the way that it does. Okay? So we want to find those genes. Now, to do that, to do this cloning, this kind of cloning, so gene cloning, it requires a number of technical things that we need to be able to do. Okay? One of those is the ability to get purified DNA. Now, what's one way to purify DNA?
Like a extraction?
Mm-hmm. Well, how do you purify it away from the other stuff in the cell? So if you find cells that have the DNA of interest,
different columns.
different columns, okay. So
like size exclusion, or like, can you run that with the DNA?
okay, or that can bind maybe the DNA and the other cellular debris gets released. So there are kits for binding purifying DNA. And this would be away from cellular debris. Now, back in the day, they didn't have those kits. But typically, these kits today that you can buy commercially, they have some sort of resin in them that will bind DNA, and the rest of the material can be washed away, and then you can elute them from that, whatever that resin is. Turns out silica is very good for binding DNA. DNA likes to bind to silica, like you would find in maybe sand. And then you treat that and release the DNA from that. So that's one way. What's another way to purify DNA that we've talked about recently?
A gel?
Yeah, gel, that's right. Electrophoresis. Now this turns out to be not very practical for lots of DNA, but it could be practical if we wanted to just get the chromosome of interest. So, not for total DNA, but if you use Chef, you could get chromosome specific. And then one other classical way of doing this, I don't know if that many people would do it anymore, but it really is the gold standard if you want pure DNA. And that is cesium chloride, typically ethidium bromide, ultracentrifugation. And the way that that works is if you take a cell extract and first you make a tube that contains cesium chloride. If you make a solution of this and you spin it at high speeds, really high speeds, like 100,000 times G, you'll create a gradient of density with increasing density towards the bottom of the tube. So the more dense molecules will wind up in this gradient towards the bottom, okay? And if you were to take an extract, so a cell extract, all the components in that cell extract will find their place of buoyant density in the tube. So in this case, when you add that to the tube, DNA will band at a particular region. So let's say that this is going to be chromosomal DNA. And that equals the buoyant density, or this spot that we're looking at, this equals the buoyant density of DNA. So all DNA in the cell, if it's the same density, will band at the same place. Okay? Now, if you add to this cesium chloride the dye called ethidium bromide. Ethidium bromide intercalates itself or gets in between the strands of DNA and binds to the in between the strands. And does anybody know a characteristic about ethidium bromide besides that it binds to DNA?
High highly
It's
toxic.
deadly.
Well, it's definitely highly toxic because it can cause breakage of DNA under certain circumstances. But why do we use it?
Is it because it's like a color?
It's a color, but specifically, how do you visualize lithium bromide stained DNA?
Via UV lighting.
That's right. So this binds to DNA, and under UV, it fluoresces. So you can see that on a gel, you can see it in the tube. Now it turns out that if you have a mixture of DNA, so this is chromosomal DNA, what if you had chromosomal DNA plus plasmid DNA? Well, if they were exactly the same density, they would be at the same place in the tube. But if you add ethidium bromide to this cell extract that contains both chromosomal DNA and plasmid DNA, the ethidium bromide is going to have an easier time getting into one of these than the other. Which one do you think it's going to have an easier time getting into? Chromosomal DNA or plasmid DNA? We'll take a vote. How many people say easier to get into chromosomal DNA? Okay. How many people say easier to get into plasmid DNA? Okay, so we're evenly divided, kind of like the country. Now, why do you think it might be easier to get into chromosomal DNA?
I mean, I was like limiting why it couldn't get into like plasmid DNA because it's like maybe like because it's in that like circle, it can't like penetrate or something.
Okay, so let's think about this. Plasmid DNA tends to be tightly supercoiled. And plasmid DNA, which is bigger in terms of molecular size? Plasmid DNA or chromosomal DNA?
The chromosome.
Chromosome's bigger. So when you break open cells, which is going to remain intact more likely? Plasmid DNA or chromosomal DNA?
Plasmid.
Plasmid will remain intact. It'll remain tightly supercoiled. If you're tightly supercoiled, is it easier or harder for ethidium bromide to get in?
Harder.
Harder. Yeah. So you're going to get less ethidium bromide. Yeah, there's a win for that team. You get more ethidium bromide in the chromosomal than in the plasmid. And because ethidium bromide goes in between the strands, so it goes in where the bases are, it's going to unwind the DNA when it binds more to chromosomal DNA than it does to plasmid DNA, which remains intact. Which means that for the same mass of DNA, plasmid DNA is going to be more dense because it's going to have a greater mass to volume ratio. The volume is going to be smaller, the mass is the same, relatively speaking. So that means that in this gradient, chromosomal DNA will be here, but plasmid DNA will be more dense. In this type of a gradient. So if ethidium bromide is there, what's cool about that is you have a way to purify, with these kits, sometimes you have a way to purify plasmid DNA and sometimes you don't, from total DNA in a cell. But using this gradient, you can get your chromosomal DNA and your plasmid DNA separately, and then the way this was done, and I don't know who still does this, but you would then puncture this tube with a syringe needle and then suck out the particular band of DNA you're interested in. So you could work exclusively with the plasmid or the chromosomal DNA source. So these are three sort of ways you can get purified DNA. Okay? Now, you have a way to purify the DNA that your source of information is coming from, so your particular gene of interest is in. Now, the next thing you want to be able to do is to cut the DNA specifically. So how do you cut DNA specifically? What can you use?
Endonucleases.
What kind of endonucleases?
Specific ones.
Okay, otherwise known as?
Restriction endonucleases.
Yes, restriction endonucleases, that's right, or restriction enzymes. Now, there are two types. You're going to get some handouts that illustrate these different types coming up soon through Blackboard. And one type is illustrated by an enzyme called EcoR1. So this enzyme has a recognition sequence GAATTC. And you should notice something about that sequence. What do you notice about it?
It's
Palindromic.
Yeah, it's palindromic. So you're going to get the same sequence going in the other direction. And the way that this works is EcoR1 makes a staggered cut. So it cuts here on this strand, it cuts in the corresponding place on the other strand. So when that cut is made in the DNA, notice what you're going to get. So this is one piece you're going to get, and then the other piece will be like this. And what do you notice about those pieces? What are they going to have at their ends?
Staggered.
Well, they're staggered, but so this piece here, just so you can see it, you're going to have the piece with the G, and then you're going to have a C, and then you're going to have the T, T, A, like that. So here's one piece of DNA, this fragment, and this fragment will have this hanging off of it. What do you notice about this relative to this piece right here? So this piece is the other piece. So now you've got the TTAA here hanging over.
They got a free place for like hydrogen bonding to occur.
Exactly. So this piece here could pair up with and bond with this piece here by hydrogen bonding. And so we refer to this type of cutting as producing sticky ends. So EcoR1 is what we call an enzyme that produces sticky ends. And that can be compared with an enzyme like SMAW1. And I believe that that particular recognition sequence is GGGCCC, which is again a palindrome. But that one cuts in the same place on both strands. So the result is that when these are cut, you get what kind of ends?
Blunt ends.
Yes, exactly. So these are blunt ends because SMAW1 and enzymes like that are blunt cutters. Okay? Now, what's important to know about this is that this allows you to take pieces of DNA that were cut with the same restriction enzyme, and it doesn't matter what source of those DNAs you have. So we could isolate DNA from your favorite bacterium that lives in the soil and cut it with, say, EcoR1. And then we could take, for example, a plasmid and cut it with EcoR1, and we can mix those two together. And because they have the same kinds of ends, they could come together and we could join them together, okay? And similarly, any fragments that have blunt ends could similarly be brought together because they would be able to at least come together because they have the same kind of end, okay? And that's one of the beauties of this, is that you're specifically able to cut DNA, and you can put together pieces of DNA that have the same kinds of ends. All right? Now, I said we could put together those pieces. That implies that there's a way to put them together. So the way that we put them together how do we typically put pieces of DNA together? How do we typically join DNAs? What kind of enzyme might do that?
DNA ligase.
DNA ligase, that's right. So DNA ligases join pieces of DNA together, and there are different types. So there is the E. coli enzyme, so E. coli ligase. And E. coli ligase uses NADH as the energy source. But E. coli ligase can only ligate sticky ends. So it has limited use in terms of this genetic engineering that we want to do. But there is a different ligase from the bacteriophage T4, which uses ATP to generate energy to make this reaction happen. And it can do blunt ends or sticky ends. So it can do both. So it can ligate sticky ends together, T4 ligase does, but it can also ligate together pieces of DNA that have blunt ends. And so typically in this type of cloning, T4 ligase is the enzyme of choice for this way of doing cloning. Okay? All right, now, in terms of putting the pieces together, if you just take a piece of DNA and you put it into a bacterial cell or a eukaryotic cell, in order for that DNA to be maintained, what does it have to be able to do?
Not be degraded.
Not be degraded. And what else?
It has to be able to join with like the genome.
Why would it need to do that? Why would it need to be able to join with the genome?
Because otherwise, like if we're speaking like for eukaryotic cells, then
It won't be expressed.
yeah.
Well, it's not so much that they won't be expressed. How are they going to be maintained as the cell grows and divides? What has to happen for DNA to be maintained in any kind of cell as the cell grows and divides?
They won't be able to be replicated.
They won't be-
cytosol.
That's right. They won't be able to be replicated because they need an origin of replication in order to replicate. And if you just randomly take pieces of DNA from whatever your source is, you're not likely to have that origin of replication there available. So you need that origin of replication, and in terms of technically being able to provide that, what we do, at least in this type of cloning, is we find a piece of DNA that knows how to replicate. What's a common piece of DNA that we're used to manipulating that has a way to replicate inside of a cell? Plasmid, exactly. A plasmid or maybe a bacteriophage. If we want to do this in bacteria, we could use a bacteriophage, which knows how to replicate in bacterial cells, or a plasmid. Okay? So we need what we'll call a cloning vehicle, which actually what we use is the term cloning vector. And we need to be able to use that cloning vehicle or cloning vector to take the pieces that we have and allow them to be expressed or be maintained inside of a, in this case, a bacterial cell, But we could also do the same kind of thing if we wanted to express something in, say, a fungal cell or a plant cell, or ultimately if we wanted to manipulate mammalian cells as well. Okay? Let's see. So I'm going to erase this stuff over here. And we'll talk about the characteristics of a cloning vector. Okay, so the characteristics of a cloning vector. So one of those characteristics we just said is it has to be able to replicate. In other words, it must be a replicon. And the examples we have are, one of them was plasmid. Another option might be a phage DNA. So for example, lambda. Okay, so it's got to be a replicon. What other aspects of that cloning vector are important, do you think?
Do you need to be able to like cut it to prepare it to receive the new DNA?
Yeah, it needs to be cuttable. And by that we mean it must have restriction sites, right? Sites for restriction enzymes. Now, should there be a lot of sites for restriction enzymes?
Limited.
Limited. Well, limited in what respect?
That like you can maybe make just one cut.
Okay, so unique, but maybe unique for each enzyme. So maybe what you want are lots of enzymes that can cut it, but each enzyme can only cut the vector once.
Okay.
So unique for each enzyme, but lots of different enzymes can cut it. So that way, let's say you got a plasmid here, you might have lots of different enzymes can cut. So EcoR1, for example, or small 1, and other ones, but they can each only cut in one place. Question?
Is this like Gibson overlap?
No.
Okay.
We're going to talk about that later.
Okay.
But so the question I want to ask is this like Gibson overlap, which probably maybe one or two of you in the audience have ever heard of. But to do that technique, which will be one of the techniques we talk about towards the end of the semester, you take a plasmid that's got restriction sites where restriction enzymes can cut, and you cut the plasmid with, let's say, EcoR1 and small 1. That opens up the plasmid, and then you can use this technique to put something into that open vector. Now, that's a modern way to do that is very efficient compared to the way that we're talking about here, but it still relies on the ability to take your vector and cut it specifically and open up the plasmid, not making lots of pieces, but basically mostly just the plasmid with very little that's been lost. Okay? All right, so you need to be able to cut your vector specifically, just in the same way that you need to be able to cut your source DNA specifically. And then what else should be true about this vector? What other characteristics? How are you going to know if it's in your bacteria or cells?
It needs to have a mechanism to be like expressed.
Okay, so it needs to be able, which thing? Your gene of interest or the vector itself?
The vector.
Okay, so when you say it needs to be expressed, what are we gonna be looking for?
The
Yes,
There needs to be some kind of phenotypic change once you've inserted your DNA or gene of interest.
okay, so that's one aspect, is that when we mix together the vector and your DNA of interest, we hope that there's a phenotype associated with that. But when you mix DNA, and we'll talk about this in a minute, but when you mix DNA with bacterial cells, does all the DNA go in? No, probably not. So how do we know which cells got the DNA and which cells don't, typically when we do an experiment like that?
we need a selectable marker.
Yeah, we need a selectable marker on the plasmid. And what's a common selectable marker when we do these kinds of experiments?
Antibiotic resistance.
That's right, antibiotic or drug resistance. Okay, so that's typically what you're going to find on these kinds of vectors that we use, especially in bacteria, but we also need something like that when we work with fungi. And even when we do experiments that involve so-called higher eukaryotes, so like plants or mammalian cells, you need a way to know that the DNA you're putting in is there, a way to select for those cells, and then after you know that that DNA is in there, then you hope that there's an additional phenotype associated with the special DNA that you're trying to clone. Okay? But here we're just talking about the vector. Now another characteristic of plasmids or phage is copy number. What should the copy number be? Should it be high? Should it be low? Does it matter?
It should be high if you're trying to clone.
Well, assuming what about the thing you're trying to clone?
That it's been correctly like integrated.
And assuming what else? Okay, so high is an option. If you have a high copy number, then your gene of interest will be replicated at a high rate. You'll have lots of it. If it's expressed, you'll have lots of that product. So say it's a protein like alcohol breakase, you get lots of that. And so that's if you need lots of your favorite gene or your favorite protein. Right? So for alcohol breakase, you want to make a lot of that stuff, you want to commercialize it, make lots of money, then maybe you're going to have a high amount of that being produced using a high copy number plasmid. But when might you want to have a low copy number? Why might that be advisable in some cases?
Precision, like if you have less, there's like less likely, like there's a less chance for like a point mutation or for a problem when it's copying.
That's a possibility. Why else maybe?
If too much of your product could kill the cell.
Yeah, what if it's toxic? So what if the product is toxic? Another possibility is if you're looking at biologically relevant conditions, so let's say you're talking about regulation, gene regulation. So again, for gene regulation, low copy number might be more appropriate. So physiologically relevant. Physiologically relevant situations might be when you would want to use a low copy number plasmid. And then there's one additional thing we want to consider. Actually, before we do that, does this all make sense up to this point? All right, so the last thing we want to consider with regard to the vector is, should it be transferable via conjugation? So if you clone this gene into this vector and you put it in bacteria, should that plasmid be able to move freely from one bacterial cell to another by something like conjugation, which is incredibly efficient if that were possible? We'll take a vote. How many people say yes, it should be able to move easily by this method of conjugation once we have it cloned in the bacteria? How many people say no, that's probably not a good idea? Exactly. So the people that developed these technologies in the late 1960s, early 1970s, they considered what possible dangers there were in using this technology. Just as now when we talk about gene editing, we're talking about the dangers, what are the risks, why would we maybe not want to just jump into that? The people that came up with this, similarly, they had a scientific meeting where they got together and said, what could go wrong with this? And what precautions do we need to make to ensure that we don't have those kinds of problems happening? And this issue about being able to transfer by conjugation, that was a big no. We don't want, once we put something into our bacteria and done this recombinant technology, we don't want those bacteria to transfer that to anybody they feel like, okay? We want that information to stay in those bacteria. We need to be able to control that and make sure it stays in the laboratory and doesn't get out into the general population of things, at least in most, if not all, circumstances. And so there are actually rules that have to be followed, at least in this country, by anybody that gets federal funding for doing research. If you violate these rules with regard to recombinant DNA work, you can have your grants taken away, and the institution you work with, if you're in an academic institution, can be barred from getting any funds from National Institutes of Health, National Science Foundation, USDA, any of those places, if you don't follow these rules. And that's one of the rules, is you do not want, unless you can demonstrate the safety of a particular instance, you don't want these plasmids to transfer. Because if you think about it, we can do this for benign reasons, right? We want to do this because we want to study the basic functions of our gene of interest, or we want to produce large amounts of this protein that we think is going to be useful to the pharmaceutical industry or to the frat houses of America. And we want to be able to do that for those good reasons, I guess. But what if we were to take the gene that codes for the ability to produce botulism toxin, and let's say we were to put that into one of these vectors and put it into E. coli, and let's say that vector could spread from E. coli to everybody everywhere else. That's a risk, right? Pretty serious risk. And so we don't want that kind of thing being able to happen. And so that's why there are these severe restrictions on the way that you do this. Okay? Questions so far? Okay. Now, there's one last thing that we need to be able to do cloning. So we've got things, all of this stuff so far is stuff that we're doing in the test tube. So we can purify our DNA with the DNA of interest that we're trying to find. We can also purify the plasmid DNA, the cloning vector. We can cut those DNA specifically using restriction enzymes. We can put them together using particular DNA ligases, but that's all still in the test tube. So there's one last thing we need to be able to do. And that is, we need to be able to put the mixture cells for expression. And the typical way that we do that is by transformation. Transformation takes cells that are made competent to take up DNA. There are a variety of ways to do that. Those include chemical treatment of the cells, another option is to remove the cell wall. And to do that, you can make what are called protoplasts or spheroplasts, where the cell wall is either damaged and then can rebuild, or completely removed. So that's another possibility. Another possibility is what's called electroporation, where you actually electrocute the cells, and that makes them able to take up DNA. And a final alternative is what's called the biolistic or gene gun approach, where you literally take DNA, put it on small beads, and you fire it at the cells. And all of these means, all of these methods are ways that people use to introduce genes into different cell types. All of these have been used for bacteria. All of these have been used at some point for different kinds of fungi. And then there's one additional way, which involves the use of agrobacterium to use its methods of introducing DNA into various cell types. So agrobacterium, did we talk about agrobacterium earlier in the semester? It's an example of a bacterium that is able to have sex with different kingdoms. So it uses conjugation to transfer DNA among different bacterial cell types, but it also normally will transfer DNA into plant cells. That's what it normally does. And in the laboratory, we've got an agrobacterium to transfer DNA into fungal cells and into mammalian cells. So it likes sex, and it will transfer DNA on its particular type of plasmid into a variety of cell types. And that's been exploited by geneticists and biotechnologists to be able to transform a variety of different kinds of cell types. Okay? All right, any questions about any of this? Okay, well, we're going to cover two more things before we finish up today's talk. What I'm going to do is give you an example of some of the vectors that are used in bacteria, sort of classical kinds of cloning experiments, and then we'll end with looking at some examples of vectors that we use in fungi. All right, so if you remember these different steps, we're going to take DNA that has our gene of interest and purify it. We're going to take our vector and purify that. You can get your vector commercially, or you can get it from a colleague who will happily send you the vector through the mail, which is okay. It's legal to do that if you get permission. And you purify those DNAs, or that comes to you already purified. You cut the DNA with restriction enzymes, and historically, when you cut the DNA, you wouldn't necessarily know where it cut. So you'd have to empirically test your source DNA to find out if it cut your gene of interest or not. So you'd have to actually, to test that out by trying different restriction enzymes and seeing it, when you cut your DNA, are you able to recover the entire gene of interest when you cut with particular enzymes? And you could try using a single restriction enzyme, or you could try using a combination of different enzymes depending upon what vector you work with. Okay? Does that sort of make sense? Okay, so we'll look at a couple of examples. When you have those DNAs that you've cut, you put them together using DNA ligase, and then you use your favorite method of transforming your cell type, whether it's bacteria or fungal or plant or mammalian, by the methods that we just talked about. So I'm going to show you a couple of examples of bacterial vectors. So this is actually an updated version. These are the PUC vectors. So the PUC series. So there's PUC 18, PUC 19, PUC 1, PUC whatever. Let's say that this one is PUC 18, if you like. This is a plasmid. It has an origin of replication for a plasmid, so it's a replicon. It has a resistance gene, so it's got a resistance to ampicillin. So cells that get this will be resistant to ampicillin. And it also has a region, so they were very clever when they designed this. They designed a region called a multiple cloning site. And this multiple cloning site, what makes it cool is it has lots of different sites for lots of different enzymes, but each site is unique for the plasmid. So if you use any of these enzymes, so let's say EcoR1, or small 1, or BAMH1, if you use any of those enzymes, when you cut with EcoR1, you'll open up the plasmid in one place. And any EcoR1 fragment could be cloned into that region. If you cut with BAMH1, it'll only open up at the BAM site, and then again, that will leave you with a molecule where other BAMH1 fragments could be cloned. But here's what's cool. If you cut with EcoR1 and BAMH1, that's going to remove this piece here, this little piece there, and the result will be that EcoR1 fragments at this end can bind here, but if they also were cut with BAMH1, that BAM end can bind there, and the result is that you get a plasmid now, where we cut here with Eco, we cut here with BAM, and your gene of interest that was cut with Eco and BAM, those hook up here, and the result is that this plasmid, this PUC plasmid, now contains PUC plus your gene of interest. So here's your favorite gene here, and it's been cloned into that site. And the other thing that's cool about this is that the way that this vector was set up is that it has the promoter for LAC-Z here, and here's the beginning of the LAC-Z gene. And this multiple cloning site is within the LAC-Z gene. So here's the rest of the LAC-Z gene. So here's LAC-Z, here's LAC-Z. But in the middle is this little place where you can clone in pieces. Okay? Now what's going to happen if you clone into that region a piece of DNA? How's that going to affect LAC-Z function?
You're no longer going to be making beta-galactosidase.
That's right. So in this case, beta-galactosidase gene, which is LAC-Z, is disrupted. So cells that get this plasmid, they'll become ampicillin resistant. So the cells, so now if we transform bacteria, let's say that the bacteria, our starting bacteria, are ampicillin sensitive, lac Z minus. If we introduce this plasmid, PUC18, into these lac Z minus ampicillin sensitive cells, what will they become? So if we take PUC18 plus the starting cells, what will they become? So if we take just the intact plasmid and put them into the E. coli that starts out this way, after transformation they will be?
They'll become ampicillin resistant and lac Z positive.
That's right. So they will become ampicillin resistant and lac Z plus. And we know that we have a way of telling whether or not cells are lac Z plus or lac Z minus, because lac Z plus cells you can grow on a certain media, and what color will they be?
They'll be blue-green.
They'll be blue-green on ampicillin plus X-gal media. But these guys, if we clone successfully our piece of DNA from that mess of DNA that has everything in it, including our favorite gene, if we clone that into this plasmid, and then we transform those bacteria, what will they become?
Ampicillin resistant, but lac Z minus.
That's right, they will remain lac Z minus. So on the plate, instead of being blue-green, they won't be blue-green. And again, this will be ampicillin plus X-gal. Okay? That makes sense? And so that's a way that we can distinguish cloning into this plasmid. If we use these different restriction enzymes, we can control the direction in which our favorite gene goes in, and we can determine whether or not that gene that we put in has disrupted the lac Z gene, because it will make those cells lac Z minus. Okay? Now the last thing that we want to do while we're working on the board is to remind ourselves that this piece of DNA may or may not have its own promoter. If it doesn't have its own promoter, so let's say that this came from a bacterial cell, and when we cut it, we lost the promoter. How can we get expression of this gene in this vector?
Through the promoter for lac Z.
That's right. So this promoter here, this allows for expression of your favorite gene. But the other thing that's cool about it is, if this gene had come from a eukaryotic cell, and even if it had its promoter, would it be useful if we're trying to express in bacteria? So would the eukaryotic promoter be of any use if we put this into bacteria? No. So even if this had its own eukaryotic promoter, it wouldn't be expressed. But the fact that we provide this promoter in the vector means that if we clone that in the correct orientation, your favorite gene can be expressed using the lac Z promoter. Okay? Any questions? Okay, we'll take a short break while we get the last slides up.